we're going to introduce kind of the themes of biology, since this is kind of a general biology class. And I also teach capstone. So I'm the only prof here mostly that teaches the capstone, which is kind of the last class that a lot of you will take. So I get you in the beginning, and I get you in the end. As a matter of fact, this year's capstone class, 5 of the students I haven't seen since 159, right, which we used to call 155, which is kind of cool to see them again. They just didn't take any of my classes. They had different, most of them were molecular biology interests, and so they weren't organismal like I am, so I haven't seen them since they all grew up on me. You know, they were little and small and didn't know what was going on, and now they're all seniors, right? So it's really neat to be, and one of my favorite things is being in this class, starting with you, and then ending in capstone. And a lot of the themes that we'll talk about today, you'll revisit in capstone and also revisit in other classes. So as I mentioned, I'm a huge photographer. This is one of my pictures from my last Costa Rica trip. So this is one of what they call the ghost crabs, or they call them red-legged crabs from the Galapagos. What's really interesting is there's a mosquito there, right? And so I don't know if the mosquito is resting or feeding on that guy between the plates. These guys are foragers. And when we get to about the middle of the course, we're going to do a really brief kind of biodiversity introduction where we'll talk about most of the major groups of animals. We'll do it quickly because there is a biodiversity class that many of you will take if you're a biology major. But we'll make sure we go through and understand some of the major groups of animals and plants as we go along. All right, so biology, right? Biology is Latin-based. You're going to see a lot of these Latin, Greek-based words as we go through here. Remember, it's kind of a different language, right, that we're going to be learning. Biology itself, if you break the word down, bios means life, logos means the study, so this is the study of life, right? And then we come to this term we call life. So what is that, right? What is life? And that's where we're going to talk about what defines life, because that's not an easy question. You know, if your parents or somebody out in the public ask you, what does it mean to be alive? You're like, oh, I know what that means, right? But it's really hard to explain, isn't it, right? What it means to be alive, or what the study of life is. Basically, biology is a science. It's an inquiry-based discipline that is based foundationally on the process of evolution, right? All right, evolution is an underlying theme. Darwin is huge, and Darwin's advocates are huge to the biological realm because all life evolves, all right? And the reason we know that is because it's been tested thousands of different ways, and over the last 200 years, evolution is real, even though out in the general public they will tell you it's not, right? And we have lots of data that supports it, right, scientifically. But to inquire is to ask questions, isn't it? So being a biologist is like being a 4-year-old. Why, right? Why, how, why? I mean, that's what we do as scientists. We're very curious. And that's how I go through life. It drives my wife insane. I'm always teaching or asking my kids questions when we're outside. And that's because I've always been that way. And so I was born to be a biologist, I guess. I like to ask questions and then try to figure out why, right? I mean, that's what we're going to do scientifically. And that's where science is. And we can do that in a couple different ways. We can do what's called discovery science, which is hard to do nowadays. We call it novel science. It's really hard to do because a lot of things are already known, right? Right? We know what a lion is. We know what, finding new species is something like discovery science. So if you were here on Friday and you talked to Dr. Milne, right, he was in here with me, he does discovery science. I do a lot of discovery science because I study the histology of non-mammalian vertebrates, all right, vertebrates that are not mammals. And for the last 150 years, nobody's cared about non-mammalian vertebrates. And so a lot of the stuff that I do is novel. We don't know. So if I give a student an organ in a frog from Costa Rica, and they start sectioning and looking at it, nobody's ever done that before. That's pretty cool, right? And they get to describe it. I mean, that's really neat. If you discover a new species, like a new spider, like what Dr. Milne does, that's what we call discovery science, where you're observing things and generalizing on the things that you see out in the natural world, and then using what we call hypothesis-based science to test whether all animals are made of cells, for example, or all plants have cell walls, right? Something that you might see in 2 or 3 plants, and then you might make that generalization that maybe all plants have cell walls, right? And that would be your hypothesis. All plants have cell walls. That hypothesis you then test, right? You start looking at all the plants. And guess what? All plants have cell walls, all the ones we've looked at. And those two are kind of interlinked. Typically, discovery science leads to hypothesis-based inquiry or science. And we'll do a little bit of both of those, right? When we start doing experiments, particularly in 165, which is the second part of this introductory course. So a lot of what I see, for example, under a microscope will be discovery science. Right? I'm looking at, for example, a sperm cell that might look really different in, say, a salamander than a frog. And then the question is why it looks different, right? And then we might make some hypotheses for that based on, which are just statements that we can test based on, maybe some of the life history characteristics of a salamander. They might live in water instead of on land, right? And that might make a big difference in what their sperm look like, for example. And so that's how a lot of science works. We're going to, at the end of this lecture, we'll come back and talk about variables and kind of how you set up an experiment and test, and you guys will get a lot of practice at doing that in different labs. And so a lot of the classes, hopefully you move into that are lab-based, that are wet labs, like cell biology. I teach physiology. I do wet labs in physiology, and I teach animal physiology, and even in my summer human physiology class, we do some, or what's called my mammalian physiology. We do some experiments that are not clinical. They're actually real biology because I'm a biologist. I'm a real biologist. I'm not a clinical biologist. Because I want you guys to understand how science works. Because even, if you go and become a doctor and want to do research at that level on humans or on tissues or on whatever it is, you're going to have to understand, kind of how basic science works. So you're going to get a lot of practice doing that. Sure, it's user error on my end. Well, I'm going to have to come down like that. That's ridiculous. All right, anyway. Okay, so another one of my favorite animals, right? So that's a snake actually my son found when we were in Costa Rica. So we were walking. This is what's called the giant parrot snake. Look at his beautiful telescopic eyes. Really cool, right? So that's obviously probably a predator, right? It's a predatory. Most snakes are predators anyway, but this particular snake keys in on fast-moving organisms. You can see it's got huge eyes. And it was telescope checking us out. It just sat there, looked like a vine, so it didn't chase us or anything. So we just took some photographs of it and moved on. All right, so we know what biology is. So let's answer this question: what is life, right? So it's a hard question to answer. It's a hard question to put into context because a lot of the machines we make today, particularly with AI coming along, right, seem alive, don't they? I mean, they have a lot of the characteristics of something that is alive. But there are certain characteristics of organisms, like the snake, like us, like bacteria, like anything that we consider a living organism, that those machines don't have. All right, and we're going to go over those as we go through lecture. And there are basically 7 of them. All right, so you can see here that we got this little robot puppy that does a lot of similar things that a real puppy does, right? Except it probably doesn't smell, it probably doesn't go to the bathroom on the floor, all of those things, right? It probably will fetch the ball if you chase it or tell it to. It'll probably interact with its environment, and it is pretty complex. It's made up of a bunch of moving pieces that work together. And so the big question is, well, how do you, what's the difference between these guys, right? And so there are things that a lot of the complex machines that we produce share characteristics with life, particularly these three properties. You know, robots, machines, a lot of what we can produce have complexity. They have lots of moving parts that have to work together in order to perform whatever function that machine does. They also move around in the environment that we have, and they also respond to stimuli. So those three properties, a lot of the things that we make share with life. And there are a couple of very important kind of scientists. This particular guy here, Ludwig Berlanthony, he was very important as kind of the godfather of what we call systems biology. All right, in systems biology, he was a biologist and a philosopher. He was born in the 1920s and wrote a book in 1968 about systems biology. And he philosophized or wrote this large volume explaining how life is a system. And this system is made up of lots of parts, right? And often what we see as a whole organism is not a true representation of all of the small parts it makes up. What that means is, if you were an alien that landed on this planet and I gave you a car that was all in pieces, you might not put it together right? You might not put it together like a car or think of a bicycle as supposed to be put together and work if you've never seen it before, right? Never seen the bike work, never seen it function. So just because I give you a bunch of pieces doesn't mean you can always put things back together the way they're supposed to interact. Does that make sense? So even though life is made up of, and even our machines are made up of a bunch of small pieces that have to interact with one another, it's not always intuitive, if you will, or straight up, we understand how those pieces actually interact and are put together. And so we call that systems biology. And this is important because often in biology, in order for us to kind of test hypotheses, we have to be simple-minded. We have to study something really specific, like a protein, right? We're looking at the protein, what the protein looks like, what amino acids make it up, and so forth. And then we look at RNA, and we understand what RNA looks like, and so forth. And then we look at DNA, we know what DNA looks like. That's what labs did, right, back in the day. And then we had to take a step back and say, well, how do all of those work together, right? So we've described the parts, kind of what the parts look like, and maybe how the parts work individually, but nothing works by itself in the body, now does it? Everything works together. And so you have to take a step back and look at the whole system in order to understand how the parts work, right? And that leads to something that's called emergent properties, which I'll talk about in a few slides. So life has been debated for long periods of time, right? And now we kind of understand, or we understand very well, because of these become major themes in biology, how life differs from, say, a machine. So these are three things that life and machines share. A few things they don't share, the other four, really, characteristics, we're going to get to in the next couple slides. So what's the difference between something that's dead, that looks like a lot of my plants often after the end of the semester, versus something that's alive, right? First and foremost, all life has lots of small parts that interact flawlessly with each other. They're integrated really nicely into a very organized system. So this is a great example. I love flowers, right? So sunflower here, picture I took at home. So this is the middle of a sunflower, and it's got this beautiful whorls of the seeds that are organized in the fruiting body of this flower. It's really organized. It's very beautiful, right? How all of the little pieces are put together and fit perfectly with one another. All life has order like this. We are very ordered, structured organisms, and the way we are put together allows us to function the way we do. And we spend we as in us, or any living organism, spends tremendous amounts of energy maintaining order. Order is expensive. All right, it's probably a term you won't hear from other professors. I'm a physiologist, so I think of things as expense. All right, and when I say expense, it's energy. We are highly organized organisms. Evolution works on reproduction, right? Who breeds with who so that you can produce organisms that are better adapted to the environment. But have you noticed that, the reproductive organs aren't really a large percentage of our bodies, are they? Right? I mean, you think ovaries are about the size, smaller than your fist. Testicles are small. That's where sex cells are made. But what's the rest of this for? And that is to interact with the environment, reach adulthood, and have the ability to reproduce, right? And so that high order is really expensive to build and maintain. And this goes against what we call the law of thermodynamics. When you get to chemistry, everything breaks down into simpler form, right? And you release energy to the environment. We have to store energy in the order of the organisms that we see. And it's very easy to see when you look at flowers, for example, or any structure within the body. So this becomes very apparent when you look under a microscope and you start looking at tissues and how beautiful they look under a microscope. Are made up of cells, and those cells are organized in certain ways. There's tremendous order to everything. Now, there's a lot of order also, as we know, to robots, right? Or computers, or whatever you think is very technologically savvy out there. So that's one thing we share with a lot of the machines or computers that our big brains can make. Also, a lot of machines and computers can react with the environment, right? Life also can respond to the environment. Now, interestingly, I want you to start thinking about the environment. When in a multicellular organism like us, there are really two environments. There's the external environment, which you're sitting in, which surrounds you. And in an animal like us, which has multiple compartments and lots of organization internally, we also have an internal environment that we have to respond to. That is, our cells have to respond to, or organs within our body have to respond to, dramatic changes in our environment, like increases in body temperature or decreases in body temperature, or increases in sugars versus decreases in sugars, increases in blood pressure, decreases in blood pressure. Those are all changes to your internal environment that you have to regulate and also respond to. But if you think about temperature is a great thing that we respond to daily, right? When you're cold, you shiver. When you're hot, you sweat, right? And that's your body's response to changing temperature, for example, externally. In this image here, we have a plant actually having the ability to respond to external stimuli. All right, so this is a Venus flytrap, a large plant that produces a specialized leaf that captures insects in particular. Some of these draw insects in by producing sweet nectar, and here we've captured a dragonfly. And often these plants that are carnivorous, we call them carnivorous plants, they live in areas that have low nitrogen in their soil. So you need nitrogen to make proteins, to make RNA and DNA. And if you can't extract that from the soil, you need to get it from somewhere, right? And so they get it often from other organisms. So they have to actually ingest other organisms. All right, so we got organization, response to the environment. Now here's one that robots can't do yet, right? Who knows if they ever will be able to do. And that is to reproduce. So all life can reproduce. It can make a copy or partial copy of itself, right? So some cells can just divide and produce clones of itself, or other organisms practice sex, right? And meet up with another of its kind and produce a individual that is offspring that is completely different genetically than either mom or dad, right? And we call that sexual reproduction. But all life, whether you clone yourself or whether you practice sex or whether you practice both, right? Some organisms do that, has the ability to produce offspring. And obviously those offspring often look nothing like the adult, right? Or they look cuter, or they look really different, say tadpole versus adult frog, than the adult does. And so not only can life reproduce, but it can also develop and grow, right? Which is the next slide. So those are two features that almost no machine can do, right? Is reproduce itself and then grow into something that is the adult form. So here you have now crocodile. It's small and cute, will grow into something savage and dangerous, right? And then produce babies of its own. And then the invention of this beautiful structure here that goes along with development, growth, and reproduction is the amniotic egg, or the egg itself, that is a portable pond where you can now take the aqueous environment that you used to have to dump your eggs into and carry it around and bury it somewhere, right? That's really how the egg works. So life can undergo development and growth along with the ability to reproduce. So those are two features that machines have a really hard time doing. Notice I'm numbering these, right? So you have all 7 of them, right, for your understanding on the exam. Life can also process energy. That is, and machines don't do this very well. You can give a machine electricity, but you can't give it gas, right, often, or you can't give it a different energy source and it can make energy from it, usable energy, right? We can do that. All right, we live in what's called an open system, and I'll show you that in a few minutes, an open system, what an open system is versus a closed one. But we eat stuff that looks a lot different than the way it comes out later because we've broken it down and use what we can to either produce new molecules or structures in the body or to produce energy. So this is a great example. So hummingbirds, all right, are often built to exploit, obviously, flowers to gather nectar, right? The nectar, or the sweet syrup that is produced here, is high in sugars, and they can ingest those sugars and then break them down to produce energy. So life can consume and then convert energy into usable cellular energy by breaking down what we're going to call macromolecules later on. Number 6, life can regulate bodily processes. And this is kind of what I was talking about with internal environment. If you want to write something down here, you can write down that life regulates bodily processes through the internal environment, right? Or what we call, a term you might have heard of before, homeostasis. Have you heard that word before? Our homeostasis means same environment, right? Or same stasis. You keep everything pretty consistent and constant inside of the body, no matter what's going on around you, right? So, great examples, temperature again. Your body temperature is always around 98.6 degrees, or a little lower than that, right? No matter what the temperature in the room is, right? If it's freezing outside, your body temperature is still 98.6 degrees. If it's 120 degrees, your body temperature tries to stay 98.6 degrees, right? Your body responds by shivering and sweating in order to keep your internal environment a consistent temperature. That is, the body not only responds to your environment, but then it regulates, right, what's going on in the body based on what's happening in your environment. Does anybody know why this desert rabbit has really large ears?
Is that desert?
Mm-hmm. Yeah, desert. Yeah, it lives out in Arizona, for example. Yeah.
It's like temperature regulation?
It's temperature regulation, good.
Blood vessels in the ears.
Yeah, great. You see all the blood vessels in the ears? So yeah, they actually don't sweat like we do. They can't sweat. So they have to regulate their body temperature. They produce a lot of heat like we do, and so they do that through their ears. Right? So that's a large surface area, send a lot of blood there. And as a matter of fact, you can actually, when they get hot, you'll see these blood vessels really easily because they've sent a lot of blood to the ears. When they're cold, those blood vessels almost disappear because they pull blood back into the body to maintain body temperature. You guys all know what toucans are? Right? Fruit Loops, right? So you know that huge beak they have? For a long time they thought it was because of what they eat or that it's predatory or that it's protection. Somebody just did a recent study that shows it has a lot of blood vessels in it and helps them regulate their body temperature. All right, it's actually really light, the beak of a toucan, but it's got tons of capillaries or really small blood vessels in it, and that's how they regulate heat, is by sending blood to and from their large beaks. So often the body regulates these processes by producing structures evolutionarily that were shaped by the environment to help them regulate their metabolism or regulate body processes. And this is a really good example of that. So the last one, number 7, is probably one of the most important ones for us biologists, and that is life adapts. All right, there's adaptation to life. Whether that's slow adaptation or fast adaptation, there are several different speeds at which animals can adapt. But the one that we're really most interested in biology is this directional adaptation. All right, this directional adaptation, which we call the process of evolution. All right, and that happens through natural selection. All right, and we're going to talk a lot about this when we get to about the last third of the semester. And so when you look at organisms, like these are all honey creepers. All right, so these are honey creepers. What's the first thing you notice about honey creepers when you look at their heads? Their beak, yeah, good. Their beak is changed, right? Their beak is very different. All of these birds are about the same size, all right, and they look really similar, so you know they're kind of related, if you will, right? Or they maybe come from a common ancestor. What's really different about them is the shape of their beak, right, which has been sculpted over time by the environment to fit a particular, we call a niche, right, or a niche, which is the niche here that the beak supplies is its feeding niche, right, what it eats. So this guy right here, nectars, right, on flowers, very specific flowers that have really long flowering bodies to get to the nectar. This one's also a nectar type of honey creeper, right, that's eating similar to like what a hummingbird would do, is going to nectar on flowers. Then this guy down here is an insectivore, so its beak is sharper, smaller, right, and it eats insects. This guy utilizes nuts, cracks nuts, looks like a parrot beak, right? The ability to crack large nuts and gather nuts. These guys are also utilize fruiting bodies, often hard fruiting bodies, in order to gather food. So these birds all exploit a different food source, therefore can survive on the same island, right? They're not all eating the same thing. They're not competing against each other. And their beak was shaped over time through advantageous mutations that happened in this group that allowed the beak to directionally change in order to feed, to take advantage of a certain food niche. So this process of evolution is seen in Darwin's finches. We're seeing this at what we call macroscopic evolution, but there are lots of small changes genetically from generation to generation that we call microevolution, and we'll talk about evidence for all of those as we move through the semester. So those are kind of the 7 properties, or the 7 characteristics is probably a better term, of life, right? So now, knowing those, it's much easier to explain if something's alive, right? Or explain to somebody what life really means. Now, because of that, because of kind of those 7 big characteristics, we can basically set those up into themes of biology that you're going to cover in a lot of your classes, right? These themes are kind of tailored from those 7 characteristics of life. And we'll come back to a lot of these in this class, and you'll see these over and over again in all your other biology classes. This is why I'm a biologist. Remember I told you I'm kind of simple-minded, I'm the dumbest person in the room probably. I like biology because it all builds on itself. That's what evolution does, right? Changes things based on what's there. And once you get a foundation of how basic biology works, then you just apply it, and it becomes easier to do kind of what they call hard stuff, like physiology. So we're going to talk about these really quickly, and then we'll come back to each of them as we go through certain parts of the class. So you can see that at the end here we have evolution again, which is the underlying theme of all of biology. So let's talk about each of these. And what you're going to notice is a lot of these are very similar to the 7 characteristics that we just talked about because they define life, right? The first one is probably one of the most important ones for me, for you to understand immediately, because it applies to biology across any of the biological disciplines. And it's called the levels of organization, or what's called biological hierarchy. And this is an old school image from a textbook a long time ago that you see in a bunch of different forms nowadays. But it shows you that biology is based on putting the parts together in a very specific way so that everything is harmonized and works together. Evolution is going to be constrained by your organization, right? That is, evolution can't change things too much. If it changes body form too much, it doesn't work very well, right? The whole organism doesn't work very well. If I make my head too big, it's just going to fall forward, right? Making my head larger, putting a bunch of teeth in it might make me a pretty decent predator, but if it's not in uniform with the entire body, it's going to cause some problems, right? You see this all the time. Evolution does this. Evolution is not God, right? It doesn't create things perfectly. There are lots of branches of the evolutionary lines of organisms that led to extinction. The animals are not here, either because they couldn't adapt or they changed too dramatically too quickly, right? And you can see some really odd and weird animals out there that, evolution was kind of, testing a form or a body shape to see if it would work properly, and obviously it didn't survive. So evolution really hones in on making us adaptable to whatever environment that we're living in. So one thing I want you to realize is that the biological realm, or what you're going to end up studying or being interested in someday, might fit into one of these categories. So for example, I live in, I don't even know what color this is. What color is that? Kind of a beige color? I don't know. Khaki color? I live in this area here, which they call the physiology realm. I'm a tissue, organism guy. So I teach physiology, I teach histology, I like reptiles, amphibians, vertebrates. I'm an organismal biologist. I know, and I'm going to be teaching you some ecology. So I've done a little ecology, but I'm not an ecologist. All right, I can teach ecology, but I'm not an ecologist. So this is what Dr. Sweets does, for example. You guys met Dr. Sweets? He's an ecologist, right? Dr. Milne does some ecology along with discovery science with spiders. The others are kind of smaller things like cells and molecules and biochemistry, right? Dr. Brunel is a biochemist, for example. So if you take biochemistry, you'll take it from him. Molecular biology involves biologists like Dr. Weber, who delves kind of in physiology and molecular and cell biology. Dr. Baker, who's a cancer biologist. Dr. Davis, those guys all do molecular biology, where they're studying molecules, what we call the macromolecules. Now, what's really interesting is that each one of these levels of organization are built up to the next level, right? So chemistry, atoms and molecules are put together to form cell membranes, right? And cell membranes form cells, right? And then cells use cell membranes, proteins, DNA, and RNA in combination to do the everyday activities in life of what a cell is, right? If you look at you're billions of cells, right, under a microscope. So those cells are not just random, are they? They're organized into tissues, right? And that's where I come in. I like studying how cells are organized architecturally, which leads to your understanding of kind of how they work in a specific location, in an organ, for example. So that's what I do. But tissues are combinations of cells that have very specific structure, right? They're organized in a very specific way. The other reason I'm a histologist is because there are only 4 tissue types. There's only one. I only have to remember 4. 4 is easy for me. I don't have to remember much. Right? There are only 4 major tissue types in your body. It's pretty amazing, actually, if you think about it. Right? You look really complicated on the outside, but under a microscope, you're not so complicated. Now, there are lots of different types of cells. Right? There are thousands of cell types in our bodies, but they're organized into 4 tissue types. And then organs are simply combinations of those tissues. So if you take a liver and cut it and look inside of it, it's made up of 3 or 4, all 4 tissue types actually, are combined to give very specific structure to the organs of the body. And then organs are combined into systems, digestive system, right? Includes stomach, pancreas, liver, small intestine, large, right? The organs of the digestive tract. All the organ systems are made up into or put together into our organism. And then organisms, if they're all the same, like Homo sapiens, like us, are a species, right? A population. And then species interact with other species in the ecological realm, right? But you can see all of these build onto themselves, right? So what does that mean? When you get sick, it might be a breakdown down here in the cell and tissue area, right? A bacteria invades your respiratory tract. It grows in your mucus, right, in the tissue there, and starts releasing all these toxins that make you feel really bad, right? Well, why do they make you feel really bad? Because these tissues are not working right anymore, are they? Your respiratory tract's not working. You're hacking. You're not, you're not getting enough oxygen. You're not feeling well, right? That means there are organs that are not working right. And if those organs aren't working right, they're going to affect other organs, right? And that is what makes you feel bad. So it's not even organs themselves that are affected in the digestive tract or the central nervous system, but it's the overall effect of the whole organism breaking down, right? Not working correctly. That leads to the symptoms of sickness, for example. So each of these levels, if you mess with one level, you're going to mess with all of them eventually, right? Does that make sense? The other thing that we'll talk about, and I think is the next slide, is form and function. But here's this is a picture out of that Campbell textbook that I showed you yesterday that shows you all the levels of organization from the biosphere, our planet, all the way down, and we're using plants here in this example, to the individual atoms and molecules here that make up these structures that are called thylakoids within the chloroplast here. That's what a chloroplast looks like under an electron microscope. And what's the importance of the chloroplast? What does it do for plants? Anybody know? Yeah.
Doesn't it like make them green?
It does. It makes them green. Good. Makes them green and pretty. I like green. I hate winter, right? When all the green goes away. All right, makes them green. Green's a pigment. So what do pigments do? They capture what? Light energy, right? Yeah, exactly. So being green, plants being green, allow them to capture light energy and produce sugars, right? So plants are the basis of our whole biosphere. Without plants, there's no life on this planet. All right, so Dr. Davis will love this because she's a plant biologist. She wants everybody to take plant biology. But we don't always give plants their due, right? I mean, without plants, there's no life. Without algae, green algae, there's no life. They use chlorophyll too. And the reason for that is because we live, or we survive, in what's called an open system. And I'll talk briefly about that in a minute. But if we start messing with any of these, we're eventually going to affect the biosphere, right? And I mean, just look around what we're doing to the environment. All of us, I'm guilty too, right? I have a car, I like electricity, I like all the modern conveniences of the world. But we are really putting pressure on organisms, communities, and ecological systems, aren't we? And they're starting to collapse. A lot of them are starting to show a lot of the stress that might be catastrophic eventually, right, on the biosphere itself. So remember that as you start thinking about things. And in capstone later on, since you focus so much on each individual kind of level as you go through your biological education, I make you come back and think about, how all these interact with one another, all these levels of organization. Okay, so far so good? Now, I was mentioning systems biology before, right? And how the whole organism is made up of a bunch of smaller parts that work in harmony with one another, right? We call it homeostasis, harmony, homeostasis, in order for the organism to function properly. So when you start putting things together and looking at all the individual parts, it's not always, as I mentioned before, intuitive how all of those fit together. Right? And that process of thinking about how all the little parts fit together and work together is what are called emergent properties. And so in biology, we often, as I said, like to make things simple when we're trying to do experimental testing, when we're trying to figure out scientifically how something works. All right? And we're going to call this reductionist view. Right? The reductionist view is I'm studying just this cell. I want to know how that cell works. I'm not thinking about where the cell is and how its neighbors affect it. I'm just thinking about that cell, right? And trying to test or understand how that cell works. Or I'm just trying to understand how DNA works. I don't care about everything else. I just care about DNA, right? That's a reductionist or a very specific view. Emergent properties are looking at an entire system is more of a generalist view, right? And so you have biologists who are reductionists, and you have biologists who are big, generalists, big theme-based people. Most of us biologists nowadays are integrative. We do both, right? We are reductionists when we're studying something we've never seen before, and then when we start to gather data from our data and from other data, we put it together to try to understand how a whole system works. And that what emerges out of that, out of the arrangement of pieces or parts of something that we're interested in, is the living system, is the system that works. And it's not always intuitive. We're often surprised. Whoa, we put all those parts together and it didn't work like I thought it was going to work. All right, it works completely different. So think about a bike. If I gave you a bike and you really had never ridden a bike or seen a bike before, it would be pretty hard to put it together so that it worked like a bike, right? I mean, you all could put it together. You all probably ridden a bike, right? But if I pulled some human out that had never seen a bike before and gave them all the parts, I mean, I don't know. It would be interesting to see what they would make, right? So it's not always easy to predict or explain how a living system works when you look at just all of its pieces. And there are great examples. Life. Life does not abide often by the rules that we see it, right? For example, I'll give you a dilemma as a herpetologist, something that we can't explain. Science can't explain everything, right? That's another good lesson you should understand. So have you guys ever seen leatherback sea turtles? It's one of the biggest vertebrates in the world. It's a huge, look up a picture if you've never seen one. It's a huge turtle. They can be, almost as big as this table. They only eat jellyfish. That's all they eat. Jellyfish are 95% water. So how does an animal that big eat just jellyfish? Physiologically, it should be impossible. There's just not enough nutrients there, but that's what they do. They also maintain their body temperature warmer than seawater. Wow. I mean, so their metabolism is even more complex than we realize. So, I mean, we're still trying to figure out how they do this. But that's what they eat. They wander the oceans and eat jellyfish, and they're this humongous complex organisms whose metabolism shouldn't be supported by that. So life is not always easily explained, even though we might know a lot about the parts of that organism. So here are some examples of things that are hard to explain as a large living entity. Things like herding, flocking, metabolism, intelligence is another one, right? Artistic creativity. How do we explain that scientifically? We have no idea, right? So those are things that we're still trying to work out. So as I mentioned, there are kind of two, and I've simplified a little bit because there are in-betweens, but there are two major ways that we can study biology, that you're going to study biology, particularly in the lab, as you start becoming budding biologists. The first one is the reductionist type of view, where we break our complex systems down into their pieces, and I study their pieces. All right? And this is what happens a lot in the lab. So I'll give you a great example of something that it was an incredible feat of human science and ingenuity and genius in the human realm. Think about the genome, the human genome. We have now basically know the entire human genome. We know the DNA sequence of all of the chromosomes on the human, in the human nucleus. That wasn't one lab that did that. That was hundreds of labs all across the world studying it at the same time over long periods of time, like 10 or 15 years, in order to sequence the entire genome of a human nucleus, of a cell of a human nucleus, right? So now we have all that information. That was very reductionist in what we did, sequence DNA. Now what are we doing with it? We've taken a step back and are trying to figure out, well, what parts of the sequence are important, right? All right, so now we went from this reductionist sequencing DNA to, okay, so what do we do with that, right? And so now people are studying where the sequences are most important. We call those sequences that we think are important, we call them genes, right? They code for something, right? They're the instruction manual for something. And so finding those genes is not easy. And so now people are focusing on the genes, right? And then once we figure out where all the genes are, you're going to take a step back and start understanding how all those genes interact with one another, right? So you can see where we're going from very specific to taking a step back and encompassing something else, right? So if then, right? All right, so we're doing what's called inductive reasoning from discovery science, like we were talking about earlier. Another great example here is the central dogma. The central dogma is the study of the interaction between DNA, RNA, and protein, all three of which were studied heavily in a reductionist sense. That is, there are protein biologists, there are DNA biologists, there are RNA biologists. And then those biologists got together collectively to talk about how they interact. All right, how those interact in the cellular sense is what is known as the central dogma. All right, it's the emergent property of those three components, if you will, of cell biology. So the central dogma, if you haven't heard of it, it's going to be burned into your brain. It is DNA to RNA to protein, right? Which is on the next slide or two. DNA, RNA to protein. And as a histologist, if I want to know if a cell is making a bunch of protein, I can do that. This cell here, this is an electron micrograph of a very zoomed in area of a single cell that's found in the salivary glands, all right, which are glands that produce a protein to digest sugars. And when you look at these cells, this right here is one little piece of the nucleus. Don't worry if you don't know what it is. So you can see this kind of circular structure. All this dark material here, we call it chromatin. Right? This is all DNA, right? So DNA is housed in a very specific place in a eukaryote, in the nucleus, right? So where all our DNA is, or most of it, not all of it, most of it, right? This cell here also has these long kind of channels, or what look like tubes, running along in the cytoplasm next to the nucleus. These long tubes are granular. You see they got little granules or little circles attached to their surface. That's called rough endoplasmic reticulum. Right, that's an organelle we're going to go over in the next chapter. I always have to do foreshadowing, right, what we're going to do next. And so all cells that have a whole bunch of rough endoplasmic reticulum produce proteins for export, right? And that's what these cells do. They produce an enzyme that they dump into the saliva that then ends up in our mouth to digest materials. Well, guess what? The protein sequence is determined by a gene in your DNA. And so that gene is on a specific chromosome. The gene is for an enzyme called amylase, which you don't even need to know, right? But it's there. They know where it is. They figured out where it is in the human genome. So it's pretty interesting. So understanding DNA doesn't necessarily mean we understand how cells work, right? It's just one piece, one reductionist piece, if you will, of the whole puzzle. So far, so good? All right. Now, systems biology is when you take a step back and you start saying, okay, now I know how all the pieces kind of work individually. Let's put them together and start trying to figure out what the whole puzzle looks like. And that's where biology's really gone. For a long time, back in the 60s, 70s, and 80s, we had a bunch of reductionists out there figuring out how everything works. And then now we're starting to put all of that information together so that we have a really good understanding of how whole systems, whole organisms work in combination with their environments, whether that's external or internal, or just how an organ works within the system that it's part of. So here's some nice questions that a systems biologist might ask. So they're not just asking about DNA, they're not just asking about how a cell works, they're asking big questions, like how does a drug affect blood pressure in a certain organ, right? That's big. That's not just looking at one organ or one body system. It's like you're giving a person a drug, what does that do to blood pressure? Either overall or in a particular organ. Or this is a really big question. How does increasing carbon dioxide affect the entire biosphere? I mean, that's a big, broad question, right? Something that we're doing, right? Dumping tons of carbon dioxide into the atmosphere, right? Which can cause the effects of global warming, right? Or at least that's what data shows. So systems biologists use emergent properties brought in, all right, by reductionists that know individual pieces and how they work. So once we know how the pieces work individually, we then put them together and study them as a unit to see if they function the same or if you got to change, how they work together as far as function goes within an organ or tissue or organ system. So that's reductionist and system biology. You'll do a little of both as you go through things. Okay, this one's huge too. This is how I think as a biologist, being a microscopist. So, structure function. Again, structure function has been shown not only at the organismal level, but at every level of biology. So, what something looks like often determines how it works. And it's been shown over and over and over and over again. So proteins are a great example of this. You can do this at any level of organization. What a tissue looks like determines what it does. I know this. I've studied thousands of tissues. So I can look at what a tissue looks like under a microscope and I can predict, hypothesize what it does, give it to a physiologist, and often it's exactly how the tissue works based on what it looks like. I can take a macromolecule, like a protein, look at its shape, and predict or hypothesize its function because of what it looks like. Proteins have diverse function because they all look different from one another. Right? So if then this is how I teach often. I show you the structure of something, and then I go into how it works, right? Because what it looks like will help you understand how it works. I'm a picture book guy. I'm very visual. So I like to look at what something looks like, and that helps me understand how it works. And this, as I said, applies to the whole organism. What you look like determines how you move and interact with your environment, all the way down to what your macromolecules look like. How you combine atoms into a macromolecule will then determine how that macromolecule functions. Right? Membranes are membranes. To understand physiology, you need to understand membranes. And one of the major things membranes do, like a cell membrane or an organelle membrane, is they compartmentalize or separate environments from one another. Right? How do they do that? It's all in the structure of the macromolecule that makes up the membrane. What that macromolecule looks like allows it to do that. And if you take physiology or you take 165, you'll learn how membranes do that. The same thing for DNA. What DNA looks like or how it's organized allows it to be a manual or a book. It stores instructions, right? And it stores information. We call it genetic information, right? It stores the instructions onto how to make proteins in the simplest sense. It's a protein manufacturing instruction book. That's what DNA is, all right, in your body, at least the DNA we're going to talk about. Now, there are pieces of DNA we have no idea what those sequences of DNA do. We haven't figured that out yet. There's always questions that we don't have answers for. Now, the cell is our basic functional and structural unit of life, okay? The smallest cells, the most simplest cells that you can find in the environment are called prokaryotic cells, right? They're called prokaryotic cells. Prokaryotes are the smallest cell that can actually reproduce and clone itself, and are a little bit smaller, in some cases much smaller, than what we call eukaryotic cells, which are what we are. We're a eukaryote, right? Our cells are bigger, and are more complicated because they have what are called membrane-bound organelles. And the next lecture, we're going to talk a lot about the differences between these two cell types. So I'll just give you a picture of these two next to each other. So these are two electron micrographs. This is a typical cell that you would find in a eukaryote like us. We're made up of billions of these, right? This is blown up really high. And what they've done to make it pretty is they colored it, right? They colored certain organelles a certain color. The real images would be black and white. That's the way I like them, but these are pretty. So this is out of the textbook here. So this is what a typical cell looks like. And you can see this is 1 micron, right? 1 micron. Now, a micron is 1,000 times smaller than a millimeter. You guys all looked at a ruler and a millimeter is like, right, like this, right? Really small, it's hard to see. So a micrometer is 1,000 times smaller than a millimeter. A typical cell in a eukaryote like us is somewhere between 50 and 100 microns, usually, So for example, the smallest cell in your body is probably a red blood cell. It's about 8 microns. Largest cell in your body is the ovum, the egg, which is over 100 microns in width. And so that's the size of a typical eukaryotic cell. If you were to put a prokaryotic cell next to it, in proper scale, this is the size of a large bacterium, blown up just a little bit. It can be a lot smaller than this. And so this cell here is much bigger because it's blown up. But you can see it's much simpler too, right? It looks like it has a very thick outer cell wall. It's got some cytoplasm. And then they colored this in purple, which is deceiving. It's just an area in the cytoplasm where the DNA is. It's not surrounded by a membrane. Where these are surrounded by cell membranes. These are compartments, right? All right, so this is the nucleus, the biggest organelle, right? These are mitochondria. These are membrane bound. These are probably vesicles, most of these that are in brown. And then these long tubes are the endoplasmic reticulum. And then the stuff that's kind of in khaki or beige color is the cytoplasm, right? So where everything is floating around. Now, all cells, it's a good test question, right? All cells, whether you're a prokaryote or a eukaryote, are surrounded by a cell membrane. So they have an outer boundary, a membrane. They have cytoplasm. So they're fluid-filled, right? Or the fluid's kind of gelatinous. It's kind of thick, a thick fluid. They both have DNA, and they both have ribosomes. So ribosomes are made of RNA and protein, and ribosomes are the workbench where you build proteins. Okay? So prokaryotes have DNA, cytoplasm, and a cell membrane, and so do eukaryotes, right? The major difference between these is membrane-bound organelles. Eukaryotes compartmentalize their cytoplasm into rooms or spaces, right? Think of it that way. And the walls of those compartments are organelle cell membranes. And this is really important because the inside of the nucleus is a completely different environment than the cytoplasm. So you can start specializing regions, right? So the metabolism, or the complexity of the chemistry, really what it comes down to, of the nucleus can be really different than the cytoplasm, right? The best example of this in a cell, there is an organelle called a lysosome. You guys all heard of a lysosome before? So a lysosome is a little vesicle, and it is a digesting organelle. It breaks things down. If you were able to stick a probe into the middle of a lysosome, its pH is 2. Right? So it's got a really acidic environment in it. The pH of the cytoplasm is 7, like most of your body fluids. And that extreme environment of 2 is only able to be maintained because of the cell membrane around the lysosome, right? Because it compartmentalizes or separates the lysosome from the rest of the cell and allows you to do that type of specialization. So organelles, or membrane-bound organelles, start allowing the cell to really specialize its metabolism and be much more complex, both structurally and functionally, right? Whereas the cytoplasm here in a prokaryote and everything else, it's all the same. The DNA is floating in the cytoplasm. The ribosomes are floating in the cytoplasm. The metabolism, though complicated in a prokaryote, is not nearly as complex as different organelle environments that you find in a eukaryote. A couple of other major differences. Obviously, there's a big size difference, right? I mean, eukaryotes are huge. Prokaryotes are much smaller. Prokaryotes have one strand of DNA. Just one circular piece of DNA. Sometimes they'll have some smaller pieces, but in general, if I generalize, they have one large chromosome, where we have linear chromosomes, and we have many of them, right? We have multiple chromosomes or multiple pieces of DNA. We have a lot more DNA. And then it's the organelles and the complex metabolism that's much different in a eukaryote. Now, one of the major functions of DNA, or what we call nucleic acids, as I already mentioned, is it's a manual, right? It's the manual of life, if you will, or the manual of metabolism. It has instructions on how to build proteins, okay? Which is going to be really important. The other major kind of, some people call direct or indirect function of DNA, is it allows for you to reproduce. Because think about it, if I just call DNA the manual of life, I have to be able to copy it to give it to a new cell, right? If I can't copy the manual, the cell that you won't have any idea, the new cell won't have any idea how to function, right? Or how to live day to day. So organizing the manual into very specific pieces of DNA, or what we call chromosomes, allows me to copy it effectively and pass it on to new generations of cells. So having DNA allows for the continual existence of life through the process of cell reproduction. Right? In the simplest form, that's called mitosis, right? The ability to divide into two cells. Or in sex cells, it's called meiosis, which is a little bit more complicated of a process. So DNA, the sequence of DNA, the ones that are really important to us are called genes. And these sequences or spaces on the DNA that we call genes, they code for something. All right, they are called units of inheritance because they have instructions for proteins, right? The ability to build proteins. So we can copy those genes, make a transcript of it, which we call RNA, and that RNA can then leave the nucleus, go out to the ribosome, and the ribosome can then read the instructions and make the protein, right? Without the instructions, the ribosome doesn't know what to do. So there are lots of genes. There are thousands, tens of thousands of genes in our chromosomes. Think about this and try to wrap your head around this. Each one of your cells, which are about 100 microns, remember a micron is 1,000 times smaller than a millimeter, has several feet of DNA in it. So you take 3 feet of thread, microscopic thread, and you put it in a cell you can't see. It's incredible, right? I mean, how do you do that? It's highly organized or compressed or condensed. I'll show you pictures of how that works when we get to talking about mitosis. So the ability of a cell to divide and copy its DNA properly is what allows life to continue, right? When you make bad copies of your genome, it leads to mutations. Sometimes those mutations are advantageous, other times they're not, right? If you forget to copy your book correctly, the new cells are going to suffer, right? They're not going to work properly. So as I mentioned here, this is a nice picture that shows here's an egg, here's a sperm, right? These are sex cells. We'll talk about how meiosis differs from mitosis when we talk about that in a couple lectures from now. But when they come together in a process called fertilization, they make a fertilized egg, something we call a zygote later. And then that egg now has part of the DNA from mom and part of the DNA from dad in the right chromosome number. It can now start to divide, right? And grow. And it'll grow into a ball of cells that eventually will grow and develop into that cute guy, right? That little dude. That will then grow and develop into an adult. The reason it can do that is because it has the instructions to do this, right? In the DNA, in the genes. Okay, so this is what DNA looks like. Again, we're not going to spend a whole bunch of time on it, but talked a little bit about the central dogma. The central dogma is DNA is copied into, or what we call transcribed, into RNA, right? DNA is double-stranded, so it has 2 strands that are linked together. It looks like a twisted ladder, right? Take a ladder with rungs and side rails and you twist it into a helix. And the sequence or the instructions are coded in the middle of the molecule in the bases that it has. And there are 4 major bases. You probably have heard of these before, right? A, T, C, and G. And the sequence of A to C to T to A to T to A to blah, 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 is actually a code for protein production, right? So it's protein instructions, if you will. We'll talk about how when we get that there. But these individual repeating units that are one of 4 letters are called nucleotides. All right, so these nucleotides are hooked together, and you get 2 strands of nucleotides connected to form a single DNA molecule. If you're human, you have 46 of these strands of DNA, right? We have 46 chromosomes. If you're human. So, and within those 46 chromosomes, we have genes, which are sequences of nucleotides that code for proteins. RNA is a copy of a gene, for example, in DNA. Let's say this is a gene right here that codes for red hair, right? This gene, in order to produce the red pigments, or the pigments that lead to red hair, we have to copy it to get it out of the nucleus, right? Because it's stuck in the nucleus, it can't go anywhere. So we do that by copying a really small segment of this long chromosome, which codes for red hair, into an RNA piece. The RNA can leave the nucleus because it's small, right? And go out into the cytoplasm where the protein will be built, right? And the protein is built at the ribosome, right? Which is out in the cytoplasm, as I mentioned earlier. So the process of DNA to RNA to protein is called the central dogma. It's called the central dogma of cell biology. You talk about it a lot when you get to cell biology. Okay, a couple more slides and then we'll stop for the day. I know I'm torturing you going over an hour this first day, right? But it helps me, stay ahead. Okay, theme number 4 out of that list of 7 is life requires energy. and the ability to do what's called energy transfer. And this is a difficult process to do. If you start studying biochemistry, chemistry, or if you love physics, and you start talking about the laws of thermodynamics, it is really hard to capture energy. It's really hard. And a lot of what you see inside of a cell is there to help try to capture some of this energy. Again, membranes will be heavily involved in this when you start talking about how it works. So, the fundamental characteristic of all living organisms is the ability to use energy to carry out life's activities. The amount of energy that is in our bodies is finite. That is, we have to collect more of it in order for us to continue to survive. That's why we eat. Mammals in particular eat a lot, right? We eat several times a day. We're ingesting components that are made up of macromolecules. Those macromolecules we can then break down into energy, usable energy that we store in a molecule called ATP. So working, moving, growing, reproducing, just surviving requires tons of energy. I called it expense, right? It's expensive to live. We have to eat a lot. Living organisms transfer potential energy or stored energy in the molecules that we eat, like carbohydrates, like sugars, into usable energy that we can use to run cellular activities. And the major organelle that's responsible for this in us is the mitochondria, right? That's a picture of what a real mitochondria looks like, right? Not a picture of one or a drawing. So this is out of muscle tissue. This is a really high power image taken under the electron microscope of a mitochondria. And you can see it's got lots of cell membrane in it, doesn't it? All that membrane is packed in the organelle. And the process of ATP production happens at that membrane. Membranes are really important. In the chloroplast we just talked about a little bit earlier, that's green, right? That captures light energy. What the chloroplast is doing is using light energy from the sun and converting it into sugar, right? Converting it into chemical energy. We can then ingest the leaves, right? Or the flower or the nectar that the plant produced, or they stored in the fruit, for example, which was captured from the sun and then incorporated into sugars. We can then eat that plant and process that energy, right? This is an open system. As long as the sun works and plants are on the surface of our planet, life will be here. If the sun stops working tomorrow, we're all dead, right? Eventually. Well, maybe we'll just grow things in greenhouses. I don't know, right? Hopefully we don't have to worry about that. So, life can transfer energy from one form to the next. Remember, energy can be neither created or destroyed. It can simply transfer from one form to the next. Here's a great image of that, right? So, you've got a primate here eating a fruit. The fruit is green. The plant that it collected off of is green. And that fruit was built by good old sunlight, right? So, the capture of sunlight through the chloroplast in the leaves allows to make sugar, which are stored in the fruits. The fruit is then eaten by the monkey, and then the sugars that are stored in that fruit are what are transformed in the mitochondria into usable energy. We call this energy flow, right? From sunlight to what's called a producer. The producers are plants, and then the consumers that eat them. We live in an open system. That is, energy is continually coming into it, right? It's not a closed system where there is nothing coming in, right? A closed system is a system where whatever is happening in a test tube that's closed off from the world, a chemical reaction, for example, is not influenced by the environment around it, right? We live in an open system where we can continually collect energy in order to transfer that energy into a usable form. And that's called energy flow. So I tried to find a funny picture because I figured about this point you were asleep, right? So organisms interact with their environment, both external and internal, right? Remember that, internal, external environments. We have the ability to sense what's going on around us or what's going on inside of us, and then respond to those stimuli, or what we call stresses sometimes in physiology. So organisms interact pretty harmoniously in most cases with their environment. Obviously, this kid's reacting to the horse or mule that came into the window there, right? Now, how do we do that? Might be the question you ask. I mean, how do we interact? How do we have the ability to sense? And it's the organization of a bunch of structures within the body that are linked together into what are called feedback loops. And so there are two major feedback loops. There are negative feedback and positive feedback loops. Okay? I think this will be the last. This will be the last slide I do. And so feedback loops work in a couple different ways. This is the most common type, the negative feedback. So if you take physiology, you talk a lot about negative feedback loops. These are loops that help keep the body in homeostasis. All right, so what does that mean? That simply means that if something happens in the body where you change your conditions, your body responds and reverses the change, right? So think about that. Reverses the change. If your body temperature goes up, what does your body do? If it goes up, you lower it, right? If your body temperature goes down, your body raises it, right? So that's negative feedback. You reverse the stress. That's the way I always think about it. You reverse the stress. I'll give you an example, a thermostat. I'll do that in a second. All right? You have a biological thermostat in your body that's part of the brain. But you know how a thermostat works, right? In your house, it's set on a negative feedback mechanism. Positive feedback is a little less common, but also very important. But instead of reversing the stress, it intensifies the stress. It multiplies the stress. So if you start a blood clotting cascade, your body, once it decides, I need a blood clot, speeds it up, right? And so you can imagine a blood clot's probably not good in your body if you don't need it. But if you get it going, it speeds it up. Birth is another, contractions. Ask any female that's given birth. Contractions get worse and worse and worse, delivering the baby, because it's built on a positive feedback mechanism. A lot of the immune system response to invaders are positive feedback systems where stress leads to more and more inflammation, right? For example. So these are rarer because they're harder to control. They're harder to control, and so it makes it pretty hard to do. Negative feedback is easily controlled, right? And so it's typically easy to control. Negative feedback happens both at the tissue and cellular level, but it also happens just in simple metabolic chemistry, right? Or what we call metabolic pathways. And that's the example I'll give you in the next slide. This is how negative feedback works right here. So you got this simple metabolic pathway where you start up here with A. Let's say you ingest A, and when you bring all of the A into your body, say it's a carbohydrate, you then run it through this metabolic pathway to produce D, right? You modify it, right? You break it down, however you want to think about it. So A comes in, and you need these three enzymes. These are proteins that take A and convert it to B, and then B can be converted into C and C into D, right? And you need these enzymes. Now, this is set up because enzyme number 1 can recognize D, okay? So when you get enough D built up, that is, you get a lot of it in your body's really efficient. It's like, I don't want to make D anymore. I got too much of it, right? And so when enough D builds up, it will shut off enzyme 1 and shut this whole thing down. And then once all the D's used up, it turns itself back on, right? Because the enzyme no longer interacts with D. It's not there anymore. And you produce it again, right? And so you can turn it off and on, all based on how much D you produce. That's typical, simple negative feedback. Now, if this is in your body, and you're thinking about something more physiologically, if temperature increases in your body, you're going to reverse it until it comes back to normal, right? When it hits normal, the whole thing shuts off. That's how a thermostat works, right? So in this room, the thermostat will kick the air conditioner on when the temperature goes above whatever the set temperature is, right? So if you're like my wife and set it on like 58 degrees, it's cold, right, in the room. You set it on 70, that's your set temperature, right? So if you go above that temperature, then the thermostat kicks the air conditioning on, right? If you go way below that temperature, it might kick the heat on, right? To bring it back to normal. Once you get to 70, it shuts off. That's how negative feedback works. Now, if this were a positive feedback loop, the more D you made, the faster you would make D, right? More and more, and you intensify the response. And that's what the next slide shows. Maybe, if I can get it to the right slide. There we go. So this is positive feedback. So the more Z you get, the more you turn on enzyme 5. And the more enzyme 5 you have, the more Y to Z you can produce. And so at the end of this metabolic pathway, you start really zooming, right? You start producing tons of Z, which might be, for example, an antibody to a specific pathogen in your body. So that, hey, I know I'm infected with streptococcus or a bacteria, so I'm going to make a protein here, an antibody that destroys it. And so once I know, I'm going to really produce a ton of it, right? This is what happens after, for example, a vaccine. When they give you a vaccine for COVID, if they were to then a week later measure an antibody for the COVID protein, the spike protein in your blood, it would be really high because you're producing a ton of it, right? Based on positive feedback. Make sense? Okay, I'll stop torturing you. Okay, so we'll start tomorrow, right here, right, on theme 6.